A good morning or afternoon to you. Thank you for joining us today. I'm Jan Reichert, Executive Director of the Antibody Society. Today's webinar is one in a series designed to inform and educate our members, as well as the broader scientific community, about topics relating to the adaptive immune receptor repertoire. The Antibody Society is pleased to include a group that works actively in this area. They comprise the adaptive immune receptor repertoire community, also called the AIR community. Our speakers today, Professor Charlotte Deane and Doctors Matthew Raybould and Fergus Boyles, are affiliated with the Oxford Protein Informatics Group at Oxford University, and they are also active members of the AIR community. Today, they will tell us how structure prediction can enhance antibody repertoire sequence analysis. Please note, the webinar is being recorded. The slides for today's webinar can be downloaded from the Materials tab in the viewer, and those questions will be answered by the speakers. Without further ado, I'll now turn the show over to Professor Deane.
Hello, everyone. It's really nice to have an opportunity to talk to you all about what we do. Just a little bit about the work that my group has done in tools for antibody structure prediction, but also function prediction and other areas around this. And I feel like I'm going to talk about something that I have been saying out loud for a very, very long time, but is only just coming to fruition, which is this idea that it's actually quite important to know the structures of the antibodies we're working with. And it's so much easier to get the sequence information that so much of the world has concentrated on the sequence information for a really long time for really good reason. We get much further if we think about structure. And to put that into a sort of very basic context, if we think about the world in terms of function, acts, that's where things happen. We need things that create a particular function. Two things are the same as far as your body is concerned if they can carry out the same function. If we look at the DNA level, and we're all really comfortable with this, the DNA level often sequences are very, very different from one another. Actually, they're the same, they might use the same protein sequence. And we can go on from that. If we look at a protein sequence, we can have things that only share very low sequence identities that have the same structure. And that follows on through to function. And so structure is much more closely related to function than the sequence information. But unfortunately, there's a big issue there. One of the reasons why we spend a lot of time thinking about sequence space rather than structural space. And that is, it's really expensive to get a protein structure. So I would love it if every time I was thinking about a particular antibody or a TCR or a nanobody, or in fact any protein, if someone would just solve the structure of it for me. Unfortunately, the cost of that, even if we're operating at really good levels, is probably in the hundreds of thousands of pounds. And that we're now able to sequence repertoires of antibodies and routinely get thousands and thousands of sequences of antibodies for any question we might be interested in, unfortunately, aren't people happily there just solve the structures for me. But that sequence thing has changed completely. I love the right, partly because I'm a computer geek as well. So the white line on that shows the speed of Moore's law. So those of you who care about computers, that is showing the doubling rate we expect for processor speed on computers. And everyone thinks this is an awesome thing, and it is, it's incredible. But just to give it a comparison, the green line going down is the speed at which the cost of the human genome has gone down. So our ability to collect sequence information is phenomenal now. So we can easily get these very large sets of sequences. We cannot very easily tell what the shapes or structures of those sequences are. To put that into a very basic antibody context, and I'm going to use my own group here, we have a database of all of the publicly available antibody structures, SABDAB, and the data graph on the left-hand side here shows structures that are currently in SABDAB. There's about 6,000 structures in SABDAB. That is all of the currently available, publicly available antibody and nanobody structures. That's quite a lot. But then we have also been trying to collect all of the available sequence data for antibodies that's out there. We haven't got all of this. And the public structures, I'm pretty clear we've got them all. But in the public sequence data, we haven't got them all. But what we have already managed to collect, we have about 3.5 billion non-unique, so redundant sequences of antibodies, and about 1.5 billion unique sequences in observed antibody space. That gap is huge. There is no way that I understand the structural space if I can only look at the cell structures. Of course, work changed in terms of whether we actually needed to solve the structures, experimentally, with the work that was done by Deane release of AlphaFold 2. I am dead certain that everybody on the webinar will have heard of AlphaFold 2. DeepMind did a very, very good job of making sure we all heard about it, and it is an amazing piece of technology, and it allows the prediction and the relatively accurate prediction of most protein structures. And of course, I could close this gap. Two quite big buts here, taking AlphaFold into the region of thinking about things like antibodies. The first one is actually how long it takes to make a prediction. So AlphaFold or AlphaFold Multima, you're talking 20 to 30 minutes on a kind of standard machine to make a single prediction. That's a pretty good computer. That's talking about, might be a bit faster, but that kind of scale. So given that scale, I can't predict the sequence. A billion would be without the computer. Anybody, and even a million is something we won't do. We might do 1,000, but we're not going to get to the scale of the sequence data we have. That's our first problem. Whatever we're going to use to close this gap is going to need to go much faster. And then the second problem is actually that AlphaFold designed the general protein prediction problem. Those of you who work in this kind of immune area are aware that immune proteins, antibodies in particular, don't behave in the same way other proteins. So the sequence alignments and the mechanisms used to build AlphaFold don't necessarily work that well when you transfer them across to the specialist types of proteins here. So our group, like many others, set about making prediction tools for antibodies, just to give you an idea of how well it works. So I'm only showing you here in that little table on the left, the RMSD, so that's how accurately we predict the structure of the CDR83, so the third loop on the heavy chain of the variable region of the antibody. And the reason for only showing you that bit is basically everything else. So the rest of the numbers are not that informative. And if it was perfect, that value would be 0. If it's anything around about 1 to 1.5, everyone would be very happy. So the first thing you can see is actually the numbers are not good enough yet. So this isn't perfect structure prediction we're talking about. We're getting reasonably good structures, but they're still not perfect. But the next thing you should see is our method is the one called AbLooper at the bottom, is that we are able to make these predictions fold, but so useful, but not perfect. But the other parts that make this potentially very useful, want to use it for, an ability to be able to predict when we've made a good prediction and when we've made a bad prediction. So if you look at the picture on the right-hand side, the y-axis of that graph is how good a prediction we have made, and the x-axis is showing you it's the RMSD between a set of predictions. So if we use it as a cutoff, we make a cutoff to say when we have good predictions. So for example, if I only take things which have kind of an average RMSD between their predictions of just 2 angstroms, I can ensure that I will make only good predictions and be able to actually give you those. And why does that matter? Because if I was going to make 1 million models and feed them onto the next stages of something, if 100,000 of them are noise, you could say, well, that doesn't matter because nearly all of them are good. But why would I put that noise in? I don't need to, because if every other model is excellent, I can take 900,000 good data points through only those and have nothing to confuse my further steps through the process. Now, of course, that does mean there might be some very important antibodies, but at least we know we can't model them instead of trying to base conclusions on poor models. The second thing that's important is how fast we can. So I've already shown you that we can make predictions about as well as everybody else within this realm, but the most useful is that we can produce the backbone conformation around 100 antibodies in under 5 seconds. That kind of changes the game. That now means if I say I want to make 1 million antibodies, I can actually go and model those kinds of numbers, so I can start to see what's possible. but I can also only use for everything I want to do next the accurate models, the ones that will be, if you like, useful for the next sets of predictions I want to make with them. Hopefully I've vaguely convinced you that it is now possible to kind of put this structural information onto these kinds of tools. And so what we want to do now is I'm going to let Matt and Fergus take you through the use of that kind of structure prediction painted onto sequence data to show you the kinds of functional inferences you can make, the kinds of things that you can learn and understand, and being able to do that. So it's over to them now.
Great. That's a relatively smooth transition by our standards. Very much. That's really the scene for the topics we're going to discuss today. So we hope in this section, which is really the main body of the webinar, to recap a little bit about BCR and antibody repertoires, why they're interesting, why we study them, and kind of what's out there in terms of data on these things. This into the traditional ways that these repertoires are analysed at the moment in the community, which tends to be a genetics-focused viewpoint, and then to try and allude to some of the areas Charlotte has introduced there to see how we can paint these structural features onto these sequences to learn different things about the data that are within our repertoires and the during kind of pathogen exposure. And at the end, there should be plenty of time for, so we do ask us Q&A as we go through, and we'll answer them at the end as best we can. So to take us back to the data here, so what is a repertoire to the Antibody Society? So I don't need to go into what antibodies are and why we're so interested in them. Clearly, they're immune proteins that can very selectively recognize pathogens and signal that they should be removed from the body. But part of their specificity is to ensure that you complementarities all exist in the body. The body actually expends an awful amount of energy maintaining a repertoire of chemically diverse antibodies. The diversities you read estimate figures that kind of depend on how much you factor in selection during development into this. But we can have a diversity roughly of 10 to the 18 different B-cell receptor antibodies based on just the configuration of how they're made up. It's an insane number. Complicated because we have different compartments of antibody repertoires. So we have the sort of antibodies and BCRs that are probing right at the start with potential pathogens, known as the naive repertoire. And then we also have a set of B cells that are preserved as B cells after exposure, sort of represent an immune history of things that were useful in the response. And then we have specialized B cells whose job is to do lots and lots of body. So within the BCR repertoire as a general concept, we have lots of individual BCR repertoires that can be distinguished by a lot of cell surface proteins and isotypes that come into in a little bit. Can also be diagnostic as to which component of this you're looking at. And they differ in physical locations as well. So BCR repertoire that's floating around peripheral blood, or you can have the BCR repertoire that's localized to a particular microenvironment, such as around a tumor or a particular site, or like the mucosal system if it's a respiratory condition. And they are constantly dynamic. So these are B cells made. This repertoire is a static, pre-coded set of antibodies that you get at birth, and that's all you've got. No, they're continually turning over, so they are responsive to the immune state that you're in. And we often see signals where they converge towards particular functions, so like specificity towards antigens that are part of a response. And this makes a really interesting study from a disease understanding perspective. And really, we want to be able to see a set of people what the BCR doing in these people at any one time. Does it look like this is a typical sort of healthy baseline response repertoire? Does it look like this is in the process of a response? And given maybe that it is in the process of response, how are these people responding to the same immune pressure? Are they responding in similar ways? And we can extrapolate these analysis of the functions of the antibodies that are going on in any one individual to study concepts such as epitope. So these are regions of antigen tend to get targeted repertoires, why certain populations might be more susceptible to disease or more resistant to disease as well. You can kind of turn that on its head. And increasingly, these repertoires providing templates for drug discovery, given that these are sort of natural produced, there's a lot of low immunogenicity that might come naturally from them. So a lot of pharmaceutical interest as well in the idea of studying these repertoires. And this whole field has called immunoinformatics, which is really starting to grow and mature into its own field as it's the computational study of the data on these repertoires. So just to take us back to where the diversity really lies in B-cell receptors and antibodies, because there are two main components of an antibody. First, the single domain, or this constant region down here. This is not really a huge source of diversity in antibodies. In fact, there are a set of 8 to 10 isotypes that kind of define different signaling abilities of the antibodies down here. Where most of the variation exists in the antigen recognition event happens are in these tips of these Ys. In your naive B-cell receptor repertoire, so your receptors that are just probing for any particular response, we have three main diversification mechanisms. Region is not encoded on a single gene. It's actually encoded across multiple genes, and these genes can be combined in different combinations, which gives us immediately there in terms of the chemistry of the binding site. We then have the events where these genes recombine to one another with additional junctional diversity. So give us length variation in some of the regions, and also sequence variation as well. We have the fact that these binding sites are heavy chain and a light chain, and so this pairing and also gives us additional diversity here. So even in the naive repertoire, there's a lot of diversification mechanisms going on that lead to chemical 10 to 10, 10 to over in terms of the specificities that are going on. And then has recognized that you migrates to lymph nodes, changes to another type of B cell, and can undergo somatic hypermutation. And there we can get mutations all throughout the sequence in the CDR loops, or the loop regions that are most proximal to the antigen, typically in this event. And this has an impact on the specificity as well. Overall, the reason these things are so diverse is a combination of these various different mechanisms. And it is this variable domain, often the FV region, that we hope when we're trying to profile what an antibody does. And so the ways we go about doing that, kind of two area. One is the this next generation sequencing approach. So this gives us unpaired VDJ sequencing. So we get that sequence heavy chain or the variable domain, and we get lots of them, up to about 10 to the 9. So we can get a very good sample of what one of the chains looks like. And we tend to go for the heavy chain because it's encoded over a VDJ combination region, so there's additional diversity, and often this follows through to a better capturing of the specificity of the antibody. We use single cell approaches. Now the compromise here is we don't get as many of these sequences, so we don't have as good a sample of the repertoire, but what we get is and the light chain. And so this data, as we see later, we can start to build full ideas of the full and structures they're in. So this is the raw data we have to work with. We often have lots, very, very deep samples of heavy chains, and we have more of these in different types of immune states and disease state. Increasingly fidelity representations of the actual antibody binding sites through single cell recognition, sequencing technologies too. And we can do strategies to sample particular parts of the repertoire, such as sorting with facts for certain antigen specificities, or we can sort for example, certain diagnostic markers to try and get the sequences of a particular BCR component as well. So there's a lot out there for these sequences of antibodies, most of it unpaired, but increasingly more paired data. And what's nice is that efforts of the AIR community to create sort of a consistent for analyses of these repertoires in a more plug and play fashion, where you know that if you have a repertoire in the database, it will have certain properties with it that will be available for you to probe in analysis. And in particular, in our tool OAS, where we have cleaned and translated all the nucleotides over to amino acids, preserve this data, you can track repertoires by individuals, disease states, ages, and time points and things. So you get a good sense of what is going on in sort of repertoire or disease associations of certain properties of repertoires. And this is quite noisy because it's an example of everything that's going on in the system, so there'll be a whole spread of antibodies that combine to different things in these databases. But increasingly, another source of raw data that we have access to are labeled antibody data. So I've shown here an example of a database we curate in our lab, which is the coronavirus antibody database. And here we have the mappings between these FV sequences and sometimes structure these FV sequences against particular pathogens that we know they can engage. A lot of this data comes from repertoires now. Of these analysis campaigns that we can increasingly avail of in our analyses. So we've got all the sequencing data that represent what's going on in someone's repertoire at any one time. So how do people start to make sense of all of this complexity and predict the functions of what's going on? We start with the repertoire data set of interest. So it might be 10 to the 8 unpaired heavy sequences, or it might be 10 sequences here. And these are nucleotides or amino acids. Most of the things I'll talk about today will be amino acids because our group works at that level, it's at the residue level. Frequently, we can get constant domain straight out of these sequences because the particular primers that we use in the sequencing can capture enough of the constant domain that we can tell what unit is attached to the variable domain. But a lot of the focus is on the variable domain itself, and the concept to map, again, to create some order to map these sequences onto predicted clones. So which sequences do we think in this repertoire are likely to come from similar progenitors? To see what sort of structure, if any, exists in the data. So this clonal mapping protocol is going through in quite a lot of detail because it's a very common technique in the field. So the first thing to do is to find this hyperdiverse CDRH3 that Charlotte mentioned earlier. So we want to find this region and take its sequence. And then the antibody that's more germline encoded, more genetically encoded, we want to map these back to whichever V and J gene they are most from. Our VH sequence down into a assignment. So we have a probable V gene, a probable J gene, and the CDRH3 sequence. This will sort of define this antibody going forward. And light chain pairings, we don't have to do it just on a single heavy chain. And once we have these representations of the sequence, the natural step is to cluster these together. It's a process known as clonotyping. And here the idea is to group antibodies with similar idea they will likely have similar specificities. So the common set of criterion that are used is to pick the same V gene, the same J gene, the same length CDRH, of a certain threshold of identity between sequences. This is typically 80%. And you can start to get these bubbles where we have antibodies clustered that we think will have similar genetics. And this is a really nice study that kind of captures all the typical things that go on in repertoire analysis campaign by Rachel Bashard-Roberts that was published in Nature a couple of years back now. And here she's analyzed what's 6 different diseases and look how just the sequences of the antibodies that are kind of drop out of these Illumina or 10x machines can tell what's going on in the response. Panels A to C here, these focus mainly on relating the isotype of the antibody, so the signaling domain, to how much of the antibodies in healthy individuals come from those signaling domains, and comparing them and contrasting them across different diseases. You can see certain diseases such as lupus has a high signal in the IgE component of the repertoire. So the IgE is often associated with autoimmunity, so that there's a lot of common sense to that. The other diseases that maybe have more viral origins have more of a mucosal IgA response. So again, just from the sequences, we can start to make sense of what we're seeing in the response. Training panels are having performed that clonal mapping. We can start to implicate certain V genes as used in response, for example, and in different components on the right-hand side here. So in lupus, for example, we're seeing an enrichment of these 434 encoded antibodies that are normally seen healthy. So again, we're seeing these really cool signals from the genetics of the antibodies that seem to be present against how much of that genetic body you typically expect to see. And then we can do statistics on the repertoire, so we can look at how clonally expanded a repertoire is. Looks at, say, the occupancy of the bubble that I showed on the previous slide. And if we have a really big bubble with lots of sequences assigned to that genetic lineage, we can typically say, oh, well, this must have been really important in the response, and we can go hunting there for things that bind to antigens of interest. We can also capture clonal diversity. So given how diverse a typical repertoire is in terms of the genetic, does this individual's repertoire seem to replicate that? If it does, maybe it's a typical healthy individual. Perhaps if there's biases here, the individual is undergoing a response, the response towards particular antigen specificities, or perhaps there's some autoimmunity or something else going on here we don't typically see. Yeah, so we can relate these expansions and diversifications to particular diseases and start to understand what's going on there. That approach to map antibodies to their specificities is to use these clonal representations against antibodies with known specificity, and it would take a very similar sequence-based clustering approach here. So we have in blue here antibodies with known antigen reactivity. For example, this C15 antibody comes from the coronavirus antibody database. We know it comes from a particular lineage and has a certain CDRH3 sequence, and we know its neutralization profile and where on the coronavirus bind. So what in this study by Galston et al, what they did was they picked the clonal clusters that were seen in multiple COVID-19 patients, but not healthy individuals, and these associated clones, and through a similar clustering approach where we look for same V gene, same J gene, and a similar CDRH sequence identity, they can start to pick out all. I think here stereotyped responses in the way people are responding to SARS-CoV-2, and we can make comments on maybe the clone based on the other antibodies of known specificity that it clustered in with. So these are really, really helpful, and by mapping the sequences of genetics and clustering, we can start to learn about what's going on in the repertoire, where's the locus of the response, and what are the specificities of the antibodies that are in the response. But it doesn't tell us everything, and there are limitations to what this approach can feasibly tell us. For example, I've picked out 3 that are made in clones. So the first thing is that you need typically to reach a threshold CDRH3 sequence identity to be assigned as likely to bind to the same target. Now, in antibodies, when you look through databases, is that the most conserved residues are actually the ones that are really the antigen. We call it the paratype. These are the residues that are actually doing the binding event. And there are certain residues in that CDRH3, for example, kind of standing by and not doing it. And then when you cluster antibodies, maybe when you think about the residues that are more likely to be proximal to the antigen, these tend to be more than the residues that are out of that. But there's no accommodating that within this classical clonal view of looking at antibody sequences. Another thing that clonotype is that the likely origin sequences must match for you to have 2 antibodies that likely bind to the same target in the same way. But you can see there are throughout IMG of technically different gene loci that have very, very high sequence identities and typically have very similar recognition genes. Because you have the same gene, sometimes you can cluster these into different sections of clonal space and actually overstating the functional of 2 by insisting that these things must match. And another factor is that clonotyping labels by the closest V gene can be a very good representation of sequence looks like. It can pretty much be entirely germline encoded. But there are other circumstances where this is a gene that's been through lots and lots of rounds of somatic mutation, now quite sequence dissimilar from the V gene. In other words, the V gene is not a good label necessarily for what this antibody looks like in terms of these somatic mutations like CDR loops can be really important for actually defining what the function of this antibody is. And so towards perhaps would be an orthogonal way of clustering antibodies together that might not be so genetics focused, it might be more structural biology focused. If we can predict residues in the different antibodies that are most likely to be involved in binding, we can allow tolerance in regions that we know shouldn't matter based upon conservation should matter for specificity, regardless of the predicted genetic backgrounds of the antibodies themselves. It's really the first place where you can help it, because it's very hard to just go from sequence to knowing exactly which residues are involved in binding to really which parts of an antibody tend to involve anti-recognition. We need these solved structures of their complexes, and after that we maybe can predict which are the residues of an antibody that are most likely to be involved in the binding event. And once we've got that, we can cluster just over those residues rather than over their genetic origin. Replace clonotyping, but it gives an orthogonal approach more from a structural biology point of view as to how two antibodies might be similar in terms of their specificities. And as Charlotte alluded to, we have a growing number of antibody structures in the structural antibody database to learn about this antigen-antibody binding event and which residues tend to be involved in it. So 75% of this data actually is useful for us because it captures the antibody in the context of the antigen. And this really has enabled lots of groups. The field leading group at the moment is a team from Cambridge, AIRR-C and AIRR-C, which is a similar method, uses a little bit more of an attention-based architecture to take these structures as input and then to output. Actually, I think these one are likely to be involved in binding, these residues in CDR2, these residues in CDR3, for example. And they do this representation of encoding of the residues that are involved in solved antibody-antigen structure, plus some expression that characterize their chemistry. And then it's trying to output a label of in the paratope or not in the paratope. So you stick a sequence in, and is actually these are likely to be in the binding. And you can do this really quite well. So an area under the PR curve is around about 0.87 for this. So this is something we can do really, really well. And based on that, then we built a tool that takes, instead of clustering over the predicted genetic, it's predict over the predicted paratope, so the residues that we think are going to be involved in the binding. Here is that antibodies that basically would be brought into the same area of function space by clonotyping. Maybe like this pair of antibodies, they have fundamentally different V genes, J genes, and CRH. When you cluster over the predicted paratope, do cluster together because they have the right kind of chemical profile that are most likely to be involved in the binding to do the same thing. And this is a real example of two antibodies, one in dark gray, one in light gray, that bind to the MERS receptor binding domain that act across the predicted together, but cluster together in a genetics viewpoint. So this is something that people could get started very, very quickly with their feeding into a clonotyping procedure is standard, and feeding into a paratyping procedure is equally standard. This is available on the OPIG resources page, and all you do is you have a like a master file sequence in your repertoire, or you have a repertoire data set straight from OAS. Paratyping feeds this into the paratype and clusters over those paratope residues and gives you lots of different cluster assignments. So sometimes these will overlap with your clonotyping experiment, kind of bolstering your confidence that yes, things have similar and they're really similar over the paratope as well. So I really think that these two things will have a similar reactivity profile. But they, for example, the example that I've shown, they can start telling different things about the antibodies in your repertoire that can't be learned by just looking at them from a genetic point of view. It's really implicitly learning structures, so how antibodies tend to talk to antigens. And now I'll pass over to Fergus, who'll tell you a little bit about how we can be a bit more explicit about this and actually predict some structures of antibodies and can help us cluster in another orthogonal way that's quite useful.
So I suppose to sort of follow immediately on from the idea Matt is espousing. So if we look at antibodies that are maybe globally quite sequence dissimilar, you can actually, if you sort of zone in on the regions of the antibody you know are actually involved in either binding or immediately around the regions that are involved in the binding, you can actually identify often antibodies that are globally sequenced but actually exhibit similar paratopes and are likely to bind to similar epitopes in their antigens. And so this is a purely sequence-based idea, but actually, before we think about sort of explicit modeling the entire antibody structure, which although, as Charlotte mentioned, we're now getting quite good at doing quickly, is still computationally expensive. One thing we might think about is where, rather than explicitly modeling my millions of repertoire sequences, can I instead, from the infer something about the structure that those antibodies or the parts of those antibodies that are involved in binding are, and this sort of stems from the observation that when you have antibodies that bind to the same epitope, to have very similar structures, and sort of particular interest that the CDR loops that are involved in binding tend to have very similar structures, and this sort of intuitively makes sense. You have sort of two molecules that ultimately want to stick together. There needs to be some sort of shape complementarity to allow favorable interactions to occur. So this is a fairly rational thing to observe. So what follows from this is thinking about how we incorporate structural awareness into the way that we cluster antibodies. And so the first thing I want to talk about is, rather than going and modeling the antibodies, can we just, from our sequence repertoire, say something just based on those sequences about what the structures of the CDR loops are likely to look like? So can we annotate a sequence with the CDR loops are likely to structurally look like this? And based off of that, can we say something about sort of the structural dynamics within that repertoire, both in terms of what is it likely to bind to, but also how much diversity is there in there? Can we infer any information about the source of that repertoire? Was it from a naive repertoire? Was it from an experienced repertoire? Something like this. So from there, we're going to move on to talking about how we can actually model the entire 3D structure of the antibody, and sort of use this sort of in de novo predicting this specific antibody is likely to bind to this specific antigen. Okay, so this idea of sort of structurally annotating our repertoires, the way we went about this process is thinking about actually what we're really interested in is what are the structures of the CDR loops likely to look like. So the approach we took the approach of two tools that we developed, one called Scallop, one called Freed. And we said, so in Scallop, what we said was the CDR loops, apart from CDR dots, were a set of backbone structures for the residues, known as canonical forms. And these loops collectively are sometimes referred to as canonical loops. And so the idea behind Scallop was to say, well, if we have all of the structural data that we can learn from. So if we take the structures do have and cluster them in some intelligent way by their structure, and then by looking at those clusters, do those clusters align with canonical forms, and based on sort of the sequences that fall into each of those clusters, can we then compare those sequences to the sequences that we find in the repertoire to say this sequence from our repertoire, its canonical loops are likely to adopt these canonical forms. And it turns out that in fact one can do this sort of very, very reliably. Thanks in part to the fact that there's a fairly small number of canonical forms that loops can adopt. And so the way we approach this is for each of these loops, so CDR H1, H2, and then the CDR loops, we map the sequence of that loop onto a likely template from a structure. And what's really cool about it, you're not explicitly doing any modeling at this stage, but if you know the anchor residues of that canonical loop sequence, you can go is not only this is the likely structure of the loop itself, but this is where it's likely to be based off of. You can annotate your canonical loops with the canonical forms, and you can also start to constrain further how they're likely set in the 3D structure, which is really cool. Now, this breaks down a little bit when we consider the CDR-H3 loop. Real sort of well canonical forms for CDR-H3, and this simply arises due to the fact that, as sort of has been alluded to previously, the H3 loop is one, it's and much more variable in length than the canonical loops, and sort of as a consequence of this, it's incredibly diverse. Incredibly diverse in sequence, also if you look in structures, those structures of solved H3 loops tend not to cluster in the same way that the canonical loops do. So we can't just map on 3 loops on canonical forms, but we do still have all of this valuable structural information available. So what we can instead do is say, can we use sort of more traditional protein structure homology modeling techniques to map our repertoire sequences onto homologue in the structural data, and then that templates something about the structure of the CDRH3 loop. And again, indeed we can. We need to do this. What it does is a two-step process of one, identifying a likely homolog and therefore template that you have a solid structure for a sequence, and then go and you actually to generate a homology model of your sequence. And so this works really well in cases where you have homologous templates available. And so what's really cool about this whole approach is even without actually doing any explicit modeling of the structure of your sequences, you can start to very rapidly group your sequences in your repertoire by their structural similarities, and then therefore likely modes of binding. And taking all this information together, we developed a tool for structural annotation of BCR repertoires called SAL+. And the basic idea of this, can we say something useful about the structural features of the entire antibody without modeling it? And so what we did in SAL+ is we combined several approaches. We took SCAP, which annotates our sequences with, annotates the canonical loops of our sequences with likely canonical forms. It uses free annotates the CDRH3 loop with a likely structural sequence, so something that is homologous in the CDRH3 loop. Then also, as a final step of this, although the framework region of the antibody is very conserved, you do have a bit of structural variety in the framework, and interesting sort of around the anchor residues of the CDR loops. And so we sort of went one step further and said, well, we can annotate the loops, but we can also actually mine structural data to assign a structural template to the sequence of the framework region of our antibodies. And so again, to emphasize, without ever doing any structural modeling at all, you're able to rapidly go through and annotate the entirety of a sequence from your repertoire with likely information about the entire antibody. Say something about what the full structure of the antibody is likely to look like without ever modeling it. Why would we want to take this into this sort of intermediary stage of analysis rather than jumping straight to full structural modeling? Well, the obvious answer is simpler and faster than full antibody modeling. So you can really scale this up to large repertoire data sets. So in SAL+, for example, we annotated at the time the contents of OS database. We're talking, many millions of sequences, which even with current advances in full structure prediction would cost an enormous amount of compute to do. But one of the other really powerful advantages of this technique is all of these methods can be applied to single sequences. So again, the overwhelming majority in repertoire data sets is still single sequence, just because that's where you get high throughput. And so you can readily apply this technique to all of, not just the paired sequences that you have available. And one of the really cool things about this that I'm going to talk about in detail now is when you can achieve structural annotation of an entire BCR repertoire, or indeed multiple entire BCR repertoires, you can then start to, when you sort of look at how they group together across the repertoires, you can start to infer things about variations between from different species or at different stages along the B cell differentiation axis. So we're going to talk in a little more detail about that now. Just to define some terminology first. If we imagine sampling data from naive BCR repertoires, we might observe, looking at how the CDRH3 cluster, actually, we might afford us, this is sort of obviously there will be more examples in reality, but imagine we had 4 distinct CDRH3 structural clusters in the BCR that we're looking at. Now, if you assumed that none of those observed structures were particularly fit, if you randomly sampled naive BCR repertoires, you might assume that each of these 4 clusters makes up around 20 of the structure of that you observe. However, in reality, what you actually observe in naive BCL is that some structure will be enormously overrepresented. So just in your naive BCL repertoires, most of the actually converge on a fairly small number of structures. And so we're going to define those as structural stems. And then you will observe some clusters that appear frequency random sampling, so we'll define that as random usage. But then there will also be some clusters that are actually incredibly underrepresented. So the and the structure are represented in the naive repertoire, but they're very uncommon. So with out of the way, one of the things that we observed when we applied this SAB plus annotation to data in the observed antibody space is when you just cluster by CDH3 template usage, one of the things that becomes immediately apparent is when you compare repertoires at different stages along the axis of differentiations, you go from naive all the way through to plasma. What you observed is, in the naive repertoires, you have massive overrepresentation of a small number of templates. And as you move sort of along the axis of differentiation, so you gain more disease exposure, you get a more mature repertoire, the pre-underrepresented templates, so structural forms, become far more common, and what you previously had as structural forms become far less common. And so this sort of relates back to this idea of your sequences undergo somatic hypermutation sort of along the axis. So as you become more disease exposed, as it matures, you gain far more diversity in the axis, and therefore in the structures observed. I think intuitively this makes sense, but what's actually really interesting about this is just by if you have some repertoire data, just by looking at how the CDRH3 usage clusters within your repertoires, you can say something about where how mature was that is your repertoire. And what's quite interesting about this also is we sort of observe similar patterns in both human and mouse. So this sort of evolution over time of template usage is actually appears to be conserved sort of across species. But we can go one step further and ask, well, other properties that we can observe that vary along the axis of differentiation, and how do they vary across different species? So again, we can entire sequence, not just the CDRH3. One of the things we can look at is how do the canonical form usages of the canonical loops vary. In particular, if you look in the naive repertoire, you will have you have a very small number of canonical forms in use, and so essentially the canonical with things that are close to the germline sequence. And again, what we observe just based off of this clustering is as you move along the axis of differentiation, the proportion of the sequences in your repertoire deviate from those of the continues to increase. And so this is another really cool way of just looking at the of your data and but something that's also quite interesting here is actually this pattern does not across different species. You see this incredible deviation from germline canonical forms, but in mouse repertoires you don't. And there's been in other studies as well. So it's a sort of a nice confirmation that what you're observing in this structural annotation and clustering approach actually matches up well about how repertoires evolve in different species. It's something that I sadly don't have a nice figure to show you, but work that we've seen recently is actually if you look at repertoires from humanized mice, one of the really cool results is actually the canonical in humanized mice repertoires. The patterns that they exhibit are much closer to those that we see in human repertoires than what we see in mouse repertoires. This is sort of a nice confirmation that actually the approach gets very close to sort of the natural behavior of human immune response rather than still being dictated by what in mouse. And so this is all sort of a really nice sort of in bulk what amount of repertoire data. But there are also some sort of immediate practical applications of this as well. And there's a really nice study in Cell from Cow et al. In 2020. So this is fairly during the COVID-19 pandemic, where they used the SAB plus annotation approach to actually infer antigen binding SARS-CoV. And what they did is they applied this structural annotation approach to paired sequences from SARS-CoV-2 response repertoires. what's interesting about what they did is the sequences within these repertoires were quite diverse, both within, but they also exhibited fairly low sequence identity to the known SARS-CoV-2 binders at the time, again, sort of in the pandemic. But under the structural annotation approach, the heavy chains actually formed a structural. And so not only do we observe that they all converge on similar cluster structures, even though they're not sequence identical, but actually this structural cluster, the loops structure, exploited a known loop template from a PDB structure for a SARS-CoV-1 binder. And so this sort of information is sort of starting to build confidence in, well, maybe actually there's some merit in some of these sequences, even though they look nothing like the known SARS-CoV-2 binders. Indeed, when this was sort of taken on to actual experimental testing, 7 of the 12 antibodies from this cluster were able to neutralize SARS-CoV-2, and they did so by a common epitope. So coming back to this idea that you can have very dissimilar sequences in your repertoire converge on common structure and a common function via their interactions with the antigen. So I think that's a really, really cool example of this sort of approach for rapidly processing enormous amounts of diverse data that actually has some immediate real-world applications. So that's all thinking about sort of using structural information implicitly. So up to this point, we haven't actually explicitly modeled the structure of any of our antibodies. We've just annotated them with look like. But as Charlotte alluded to earlier, the use of full structural it just continue to grow and grow and grow in terms of popularity, in terms of the scale at which we're able to apply it, and in terms of the accuracy with which we're able to model antibodies. And as we increasingly have access to paired FV repertoire data, one thing we might start to think about is what is the best way to actually go about large-scale modeling of complete structures. So as Charlotte sort of alluded to earlier, something such as AlphaFold is an incredibly powerful tool for prediction of protein structures in general. It's perhaps not the best way to approach antibody perspective, but you still want to achieve high throughput. So there are several different ways of sort of doing full structure modeling for antibodies. The way we sort of approach this in the group with our tool ABody Builder is to sort of think of it as sort of, again, going back to this idea that we can split up the structural annotation of the antibody region, canonical loop, CDRH3. We can do exactly the same thing when we want to model the antibody. So rather than just feeding it into a full structure predictor, what we can do is we can take a framework region, mine the structural to identify homologs that we can use as structure templates for the framework region. We can then find some structure templates for the canonical loops, which we're very confident that we can do because you don't have very many canonical forms. And then you can look for the CDRH3 loop in all the structural data. And if you happen to have a sequence homolog for CDRH3, you can use sort of template-based modeling for the CDRH3 loop. Have such a template, then you can always fall back on ab initio modeling. So you can use standard tools such as Modeler for this, or what we have a tool called Links, which at the expense of increased runtime was able to sort of generate more accurate antibody CDRH3 loops. And once you have all of that in place, then you can think about modeling side chains. And so by approaching it in this manner, you can achieve sort of quite good accuracy, both the canonical loops and also acceptable accuracy in the CDRH3 loops. And the real advantage of this approach is you could model a full structure on the order of 20 seconds on a single CPU. So we're getting closer to that sort of high throughput scenario. But earlier, sort of something that's revolutionized protein structure prediction in general recently is the use of deep learning, and in particular, the use of equivalent graph neural network. Wow, AlphaFold really. And so we sort of thought, well, okay, this works for general protein structure prediction, but so can for antibody loop prediction. This is the part of antibody structure prediction. And so we developed the tool AbLooper, which uses any to predict the structure of all of the antibody loops, both the canonical loops and the CDRH3, starting from just the antibody sequence with the constraint of knowing where the anchor residues are. So as Charlotte sort of mentioned earlier, this method proved to be very, very fruitful, and so it could both rapidly and accurately model all of the loops. But something that's actually important to note is that the most accurately modeled is not necessarily the most useful. So what's often required is actually when you model each CDR loop in isolation, you get a very good model of. You actually put that together with the other loops and the rest of the antibody structure, and potentially have it binding with an antigen. You actually need to go and do some further modification in the form of energy minimization, piece all that together into an actually useful antibody structure. Again, something that Charlotte mentioned earlier is about this is, and it sort of stems from the fact that you're no longer doing sort of template-based modeling, is by the very nature of this sort of ab initio approach, you have diversity in predicted loops, and therefore what you can do is you can say, well, I have 5 different models for my loops, and they're all really, really diverse. So I have lower confidence in whether or not I've actually gotten the model right, because none of my models agree. 5 models and all agree, even though it was an ab initio approach, you can be reasonably confident that you're likely to have converged on a sensible solution. So just to reiterate sort of what Charlotte said earlier, some of the advantages of the ab-looper approach is that, so compared to the way we were modeling antibody loops previously, you can sort of reliably sub-angstrom accurate canonical loops and to within about 2.5 angstroms on CDRH3. And again, you sort of have this estimate of confidence. So you tend to get accurate predictions, but you have a way of saying when you suspect you may not have an accurate prediction. And so the advantages of these sort of deep learning approaches is if you have the hardware available, you can take full advantage of GPU acceleration. And so you can model hundreds of backbone conformations for your loops. And so, for example, from a 10x phone can model less, which is a really, really cool breakthrough. And so if anyone's interested, the ab-looper code is, and it will be on our website soon. So I'll hand you back over to Matt now.
Yeah, brilliant. So we've sort of seen there how structure can be used implicitly to predict and cluster based on which residues are most likely to be involved in binding. We've seen how we can apply structural annotation to isolation from one another and cluster their predicted likely structural properties and learn what's going on in the repertoire from that. Are any of these clusters of similar loop structures? And now Fergus has walked us through actually how we could go about building full FV models of our paired sequence data. And so really in the next few slides, I'll just show we think really cool applications of what you can do with these full 3D models of the FV binding site. And another interesting thing to note is that for ab-looper as our way of modeling antibodies, because it performs better than a bodybuilder, a lot of the stuff that we've been able to inject so far has been more classical homology modeling approach. And so we're excited to see what even better structural models can do to our interpretation to get better representations. So this first idea is if you have a set of FV data for which you say know what the antigen is, or you are highly these antibodies probably bind to similar targets, so that you can get this data, for example, by antigen sorting from a repertoire and then sync. But with this data, one thing that we really want to know is which of these antibodies are likely to bind to the same epitopes. And this is important because if you want to, for example, design combination therapies, you don't want to include 2 examples of antibodies that are likely to compete for the same site. We would like to pick antibodies that go for different sites and things like that. Epitopes are better for in the context of viral infection, and so not all epitopes are made equally. And we really want to be able to start to make commentary about not just that this antibody has drawn down into the antigen. We want to know some order within that as to how they're, the similarity antibodies are likely to have in terms of aging. And using this idea that antibodies with similar structures tend to bind with similar binding modes, we've developed this space tool, which is structural profiling of antibodies. And it's a very simple concept. You just take your antibody sequence data, and here we use the sort of artificial sorting approach by taking the database. So we know all of these antibodies bind to coronavirus antigens. At the time, we had around 2,000 data points. We have closer to 4,500 of these now. But we fed them into the structural modeling, the FV structural modeling process, filtered for the set that we think are modeled most accurately, and simply clustered them just on their backbone topology. So no chemistry going on here, literally just saying which antibodies have similar shapes to one another. And we have to look at the metadata that's associated with the sequences we put into the same structural clusters. The vast majority bound to consistent domains. So we were able to use antibodies in a sensible way, given what we know they bind to, just from the FE model, literally nothing else. And quite a high portion of the antibody data too. And indeed, some where they disagreed, maybe on the metadata, could actually be linked back to experimental threshold. In the alive, say, for example, couldn't quite delimit whether it was an RBD binder, an NTD binder, and things like that. So already getting very high performance by just thinking of the shape of the antibody and grouping things by that FE shape. And here is a sort of visualization of an example of 70 antibodies that we modeled. This demonstrates really what you can do when you have a hypothesis of how some antibodies combine. For example, a solved structure of an antibody that's already bound to a coronavirus antigen. Alignment-based docking approach, where you take the models of the antibodies that have been grouped into a cluster, a structural cluster, and you combine those with the one example structure, and you can see that all the antibodies within this cluster seem commensurate with a particular epitope, a particular way of approaching the antigen. And this allows you to do epitope profiling, and to really not just say that these are likely to bind to the same epitope, but which epitope they're likely to bind to. And from the CDR3 and CD3 sequences represented across these antibodies here, we can see it's a huge diversity of sequences all linked to the same epitope. So we can imagine a kind of expanded clonotype where we don't really care who had an antibody in this clonotype, 2 of 10 had one in the second clonotype, and 2 of 10 had one in the last clonotype. Fundamentally, the same antigen and specifically to the same epitope. And so these are the sorts of signal we're able to draw from the data by considering what we think they look like in 3 dimensions based on that. Oh, sorry, just on that last slide, the summary there is that start to pick out actually that already we thought the response to SARS-CoV-2 was quite public based on that work from Galson et al., where we just literally did the genetic clustering some maps with other known repertoires. But when we start to group together genetically dissimilar things that can do the same function, we see it's even more public than we thought by phenotyping. And the amount of pressure on particular parts of the SARS-CoV-2 spike protein is being borne out now with these variants that crop up in quite predictable ways. And interesting, by considering structure rather than being hyper-focused on genetics, is that we can look at similarities between different species. Now, focus that really nice example earlier of chemical form usages and deviations across different species and how they have different properties. But actually, think about it, virus is equally foreign to us as it is to mice. And when you start to group these antibodies together by structure and not sequence, we can see epitopes that both humans and mice fail to distinguish the antigen as not self. So you'd never ever be able to cluster the genes of the mice and the humans together to find which genes can do similar things. But if you think about their structures, sort of mapping it into a different part of the space, you can start to make functional inferences of how humans might respond similarly to similar targets. So that was just literally just using the canonical, well, just literally using the back prediction of the FE domain, but sequence of the antibody, so we can use some of this information as well. It's not off limits to us. And this is really the idea behind the AppLigity software. I showed you paratyping earlier, which was kind of purely sequence-based and learned implicitly from the structures of antibodies, talking to antigens to work we care about. But with a full FV model, you can put those residues I should care about onto an ID structure antibody and a rich representation of how these residues might talk to one another in terms of interaction groups, not just the consecutive sequence, because frequently residues that are in sequence fold together when you consider their structures. And so we have this relative spatial positioning of the paratope residues can be quite hours to draw, again, thinking from a clustering perspective, functional inferences of antibodies that might have not looked similar just literally in terms of the similarity of the paratopes, but actually a subset of the way that these residues are connected in space brings them towards, well, actually they might be able to bind to the same. And so this was the idea behind obligity, where we represent these antibody binding sites by the color of the chemistries of the different residues, but also the spatial positionings. And we represent this via what are known as hash tables. And so we can put these sort of triangular representations of different antibodies, these different residues on the antibody might talk to one another in the context of a binding site. And so we map this sort of spatial paratope into and use methods of similarity between hash tables to cluster these representations together. And based on a threshold that's been set on solved structures that we know bind to the same antigen and kind of the properties of their paratopes and what are the conserved features there, we can set a cutoff. Say if your Tversky similarities, we're going to group these antibodies together as potentially able to bind to the same epitopes. And these are two really nice examples of how this can take you even further beyond what paratyping can do by considering the structure. Here we have 4 antibodies, 3 of which will engage the same antigen and 1 of which does not engage the same antigen. So it's fundamentally different in terms of its specificity profile. If we just look at these antibodies based on their CDRH3 profile, all of them would be scored as dissimilar. So these are not going to get clustered different in that way. If we think instead by obligity, we actually are able to surpass the obligity all of the antibodies that engage the same antigen, whilst retaining the sensitivity of finding the antibody that doesn't engage the same antigen, that doesn't meet the obligity threshold. And so what we're showing here, the paratopes of these antibodies underlined in bold here. Sometimes the paratope identity would fall below clustered together, I think, clustered together. But because the spatial positioning of certain key residues across the interface is similar enough, we group together the antibodies likely to bind to the same site. So we can take even bigger jumps in genetic space, in paratope space, to spot the functional commonalities between antibodies with as well. And this allows us to play around with ideas that we don't even necessarily need the same length of CDRH3 sequence. So long as maybe it's the same structural positions, we can have antibodies with different CDR lengths that might bind to the same site. And here, antibodies where the same shape for the H3 can adopt 2 different shapes and bind to the same target. And obligity was able to sort of see beyond the fact that these antibodies are different H3 loops and see there's enough similarity here that they're likely to bind to the same site, which is really cool. Another application of models is that we can start to look for how we define the structural diversity of an entire repertoire. We've looked in isolation. What we want to see is all the FV shapes that are involved in my repertoire. So this sort of flow chart on the left side shows one way to do this data, where you have to pull together heavy and light chain sequences and consider how they might pair with one another. But increasingly, we have this pairing from 10x data. We can do this easily, where we just take the paired sequences, model them, and then we can look at the diversity of these models and compare where these models sit with different individuals. We can go from sequences to snapshots of the FV structures that are present in someone's repertoire. And as we different individuals to look at how similar the structures that are sampled in their repertoires are to one another. And we end up with these ideas of more private structures that we see in a subset of the individuals, public structures that are observed in every single individual. And what we see in the genetics world in terms of how similar people's repertoires is that actually most people's repertoires are clonally private, right? There's this beautiful study from Brian Briney in Nature where they did really, really deep VH sequencing of individuals and found that just 0.00% clonotypes were shared across 10 individuals, which would imply that if you had to belong to the same clonotype to respond to the same antigen, we shouldn't be responding to the same antigens in the same ways. But actually mapping this onto structural space, given that structure is often also conserved across same antigen binders, we actually see that of the structures we persist even when you keep adding new individuals. So about 3% of these distinct structures we see in everyone. And this is consistent with the sort of overlapping reactivities we see between people's repertoires. And we've proposed this as a structural kind of basis set. And we've got ideas that maybe we could use towards library design for drug discovery. We've also taken this set as representative of what the antibodies typically look like in humans to learn about maybe the therapeutic antibody profile. And so we're playing around with this idea and what we can learn from these really common 3% of structures. Also is that are quite similar to the clonotyping idea. So we mapped the common FV structures that are present in 3 individuals before vaccination, and then look at how those commonalities change after vaccination. What we see is that the proportion of structural similarity in the repertoires goes up after vaccination. And this might implicate particular structures in the actual response, for maybe the changing of canonical classes that you don't typically get at baseline, but you do get after antigen responses being seen here. And so this is another to study and learn what this can tell us. And then finally, Fergus will wrap this presentation up with an idea of we can do an antigen in mind, but we don't maybe have a lot of examples of antibodies that actually bind to it, so cluster so much of an option. We might have to think about this in a different sense. This is very early stage work, but we're thinking in this area, and Fergus will sort of walk us through how we're thinking about it.
So this sort of final idea is, I think it's going to probably sort of this part of this presentation, as Matt said, it's very early stages research. So the idea, and it's sort of limited by, in order to start thinking about sort of analyzing the library to try and identify sort of novel binders for an antigen of interest, and one must first construct a reliable antibody. It's a non-trivial challenge. Even in the advent of increasingly available paired data. So one further I have a library of structures available, let's do something cool with it, is I might say, well, I have a repertoire associated with the disease. Topical example obviously being SARS-CoV-2 and sort of related coronaviruses. I might immediately ask the question, does this repertoire contain something useful that we're interested in? So for example, I have a SARS-CoV-1 repertoire. Is there anything useful in there for SARS-CoV-2? Yeah, we have this library present. We have the ability to go and model. We can sort of cluster and then model a subset of this library to get sort of some set of representative structures to represent the diversity in my repertoire. Can I come up with a way of randomly screening? And I come from a small molecule drug discovery. With the small molecule world, the idea of I have a library of compounds, I screen it against a protein target of interest is old hat. People have been doing it for a long time with varying degrees of success, I must admit. But can I come up with a method, given all of the structural data and a solved structure idea of my antigen, the method of actually screening this library of antibody model, things that are likely to actually bind to that antigen target? And often this is option that you really have if you don't have some sort of prior data or examples of antibodies that engage your antigen of interest. So there's already sort of been a question about sort of a truly novel target in the chat. So you have some repertoire for maybe a related disease, but no knowledge of known binders for your new target. Maybe this is probably the best place that you can start. And so the way we approach this is sort of taking ideas of what people have done in the drug discovery world, where people found that with the ability to represent a 3D protein structure in a way that is amenable to being fed into previous convolutional neural networks in sort of analogous ways to how they analyze 2D images and 3D videos. So you represent it as a set of images, sort of take each atom or atom type in your structure as sort of a different channel analogous to color channels in images. So one image is actually 3 different sets of pixels, one for red, one for green, one for blue. You can do the same thing with protein structures, feed that into a convolutional neural network. Had sort of a lot of success in a small molecule will bind to a protein of interest. So we thought, can we do the same thing with antibody antigen binding? So can we train a neural network to say, based on structures of antibodies and antigens, to say this antibody is likely to bind, this antibody is not likely to bind. And one here is sufficient to simply have a structure of the antibody and a structure of the antigen, because that you still need a way of saying this is the epitope, this is the epitope, but also this is the 3D confirmation. We think they would bind if they bind. So this is our best guess for how they fit together. And so this whole process necessitates, which, if you're familiar with it, is basically the idea of I have one protein, I have another protein, I rotate and space, try and predict how they will sit together. But given that you're able to perform that to some degree of success, you can then do these things bind. And so we did this in our tool DLab. And so just a very brief overview of this pipeline, because it's really sort of a multi-stage process. You can't just go from sequence to predict bind. We start by saying, okay, let's use a bodybuilder to model the structure of our antibody, and we have a crystal structure of our antibody target. And then we use tools such as ZDock to predict a set of docking poses. We use an ensemble of 500 poses, for example, for this is how we think things could fit. This is their 3D complementarity. And then it turned out that it wasn't sufficient to just go from that to immediately predicting the binding, because when you do docking, if you deal with it, oftentimes you're able to sample the correct binding pose, but it's actually really quite difficult amid all of the incorrect binding poses that you sample to pick out the correct binding pose. So it turned out it was actually first necessary to train one convolutional neural network, DLab-RE or DLab-RE score, to actually take the list of poses out of your docking tool and predict this is the one that is actually the best pose, rather than just taking the one that the docking tool thinks is the best pose. And so from that, you can reduce it down to poses by thresholding that at some level of confidence, or taking the top n%. You were saying earlier about not to feed noise into your model necessarily, you can arrive at sort of your best chance for predicting binding. And then given this, you can then take a set which is trained in a slightly different way for a task, DLab virtual screening or DLab VS, to predict all of these things that I think I have a good guess for how they stay binding, or it's just some sort of spurious 3D shape complementarity but can't form any interesting interactions. Given that the correct true binding event. And just to very, very quickly give you an overview for sort of how well this performs. So the figure on the left here shows how well you so the first set of bars shows how well you would expect to do if you randomly sampled from your library of antibodies. So the blue bar shows the ratio of the chances of having a binder in what you've sampled, 2%. The red bar shows the likelihood of getting a true binder if you take the top 20% of things in your library. So there's obviously, when I do virtual screening, I'm trying to reduce a big library down to a tractable number to test experimentally. The percentage, the top n% that you can take is going to be dictated largely by how much stuff you can, but clearly has an effect on your ability to pick out a binder. And so what we found was when you lab VS and use ZDock to predict poses on models of your antibody structures without doing this thresholding of sort of removing things that look like bad dock poses, you are able to enrich your hit rate for picking out binders a little bit. It gets about maybe twice as good to around twice as sampling your model line, but it's not great. So there's obviously this issue of if the dock pose is bad, you can't really predict if they bind. However, when you apply this additional thresholding, the DLab-RE method to pick out good poses and throw away things where you think it's not such a good pose, you can increase your enrichment rate to maybe 4 or 5 times the likelihood of randomly pulling out a binder from your library, depending on how much of that library you can sample. And on the right, it just shows when you look at when you have different amounts of different numbers of poses available. When you start thresholding at different levels, you're far more likely to, and this is also very intuitive, you're far more likely to actually pull out a pose that is quote accurate. So you're filtering out a lot of noise by having a reliable way of doing this thresholding. And so it is early days, but this appears to be a potentially very powerful approach of interrogating the repertoire data that you have available when you're trying to attack sort of a truly novel antigen for which you don't necessarily have any known binders as a starting point. Just to wrap up, sort of with a very high level overview of lots and lots of different approaches, ranging from sort of just exploring things in sequence space right the way through to full structural modeling. With sort of advances in availability of paired data, plus the accuracy and speed with which we can reliably antibody structures, there's a growing role for different forms of structure-based approach to repertoire analysis. So you start with this idea of just annotating structures or predicting paratopes, through to trying to cluster things in structure space, through to actually explicitly directly trying to predict sort of binding dynamics, binding events, just purely based off of those structures with no prior knowledge to exploit. So if anybody's interested in experimenting with any of these tools, maybe applying one or all of them to some of your data sets or workflow tools, paratyping, SAP+, Scallop, Ablicity, and DLab, are publicly available on our GitHub page, OPIG. They're all available. You can go download them, have a play with them. The SABDAB and OAS databases, as well as the derivatives like COVABDAB, TheraSABDAB, are hosted on our website. So you can explore the data in those. All of the SABPred tools, such as a body builder, are also hosted on our website. So you just have a sequence of interest, you want to generate a model for it, you can use our website. But if you would like to get hold of the SABDAB database and the tools such as a body builder that are dependent upon it, we distribute the entire platform as a virtual machine and as a singularity container called SABbox, which is available through the Oxford University. Links on the slides. It's available completely free of charge for academic use. So you just get a hold of the container or the VM, download it, unpack it, and go and play with all the tools. There is a license fee for industrial users, but this is continually updated with any new tools. For example, ABLOOP. So you have continual access to all of these tools. So with that, and with the knowledge running on, so we'd just like to thank all of our industrial collaborators, all of the members of OPIG who have been involved in the development of many of these methods, as well as some of the current members of OPIG who are continuing to drive forward the development of some of these methods. So this is the entire group here. I was ill, so I'm not there, sadly. So thank you very much for listening. We're aware that in our covering topics, but in the time that's remaining, we would be happy to answer any questions.
Thank you very much. We do have a few questions, so let's just get right to it. Is there any threshold regarding CDRH3 length and accuracy to structure?
To some extent, so I suppose it's important to template-based method and ab initio methods. Obviously, when you're doing template-based modeling, for example, what we would do with Freed, you're less constrained by is less influenced by the length of the loop itself, and more by whether how homologous the best template finding is. So if you have a really long CDR, but that happens to be a really, really good template in the known structural data, chances are you can generate a really, really accurate model even with CDR. However, obviously, in the upper, a very small percentile of solved structures in terms of length, you're less likely to have a good template. Your model have a bad model, but that's more just the consequence of it being data driven, rather than the length itself. Methods, and for example, ABLOOP is completely length independent. There doesn't appear to be an enormous issue in terms of sort of slightly longer loops being hard. But just in general, a longer loop, it will take longer to model. And depending on the method you use, yes, there may be some issues with accuracy. It's not quite as simple as loops above this length are harder to model in that respect.
Next question, is it reasonable to assume that structure of the CDRs is related to the solution it is crystallized in, and the structure in, say, PBS would be different?
I think in general, the thing we have to remember with all structures is that they're measured in some way, right? And they're often measured in a way that it's quite destructive to the protein that's being assessed. So if we're talking about exocrystallography, we're bombarding it with X. We're talking about we're freezing molecules. So we are perturbing the molecule during the structure solving process. And so yes, it's important to consider thinking about this very rigorously, that any way of solving the structure is like to have an influence on the structure we actually see. And this, most of the structures in SABDAB, when they're X-ray structures, are similar ways, and so we assume that in the context of all the noise that's going on here, this is one bit of noise that can be kind of disregarded. But yeah, theoretically, it should happen.
Next question. Since the HCDR3 has too much variability to organize into a set of canonical forms, but still is most important for binding, what kind of information can you get from these canonical forms for other CDRs? And equivalently, may the epitopes be classified into canonical forms, or, as for HCDR3, is variability too great?
So I think there's a few parts of that question that kind of need unpacking, and I think this was asked actually before Fergus walked through the canonical form clustering with things like that. So clearly we can learn a lot from the canonical forms, even though classically they are considered important for binding site. But the idea that CDRH3 is always more important for binding is a simplification. It tends to be, but again, it's not in certain antigen contexts. It isn't the dominant rather than binding. Actually, the other CDRs are much more important. You can imagine very short CDH3s that don't have quite featureless, maybe they're mostly glycine containing. They're not going to play a huge role in the binding. So first of all, yes, it's very important, but the CDRs also are very, very important too. And the point about epitope prediction is a good one. So we still are very predict an antigen, what is the epitope of an antigen. So we don't really know the features of epitopes. So while it feels like there ought to be some classification that we can group epitopes together via their canonical structural features, right now we're not at the point where we can do that. In recent approaches where people have tried things like antibody fragments into antigens and using that as a vector for which epitope similarity in terms of antibody, but it's still a very early days for that field. So I think this is something that people want to quite do. Yeah.
Next question. I think this will be a quick one. Perhaps I missed it. Is CDR being defined by IMGT in all of these studies? If yes, CDR-L2 is only 3 residues, which might not capture CDR-L2 accurately, perhaps. If not, which definition is universally or accurately recommended for paratope and clonotypic analysis?
So very quick, use the North definition. And the reason we do that is because that's more of a structural definition of the CDR rather than genetic definition of the CDR. IMGT is really, really useful when you're comparing antibodies to TCRs or antibodies and nanobodies or different classes of immunoglobulin, because the equivalent regions have roughly the structural position. But if you want to say stick together an antibody via a modular approach, it's better to use a definition of the CDR that captures the integral element. And that's why we tend to use the North definition. And in the North definition, CDR-L2 is 8 residues long. There are lots of analyses where I think that maps where they've talked about the impact of using different CDR definitions on your conclusions. Yes, that absolutely does make a difference. But as model, since we have a principal use definition based on the modularity of the structure, so that's why we made the decision to go.
Next question. With the deep learning approach, how would you expect structural prediction to behave for a new or less well-studied target like a novel vaccine antigen target?
So that's a really good question, but I think, and this perhaps doesn't go much, but also there are similar issues with sort of previous template-based approaches, or even any other data-driven ab initio method. And what you would really expect is if the antibodies that are like a novel target are sort of incredibly sequence dissimilar to anything that you have in the known data approach, or template-based or database search method, is going to be, if not going to, then at least is going to have much less data that's learned previously to draw upon, and so either will be able to model it less accurately or confident in its prediction. For example, if you think about blueprint, it might be more variable in its predictions because it's less constrained by what it's seen previously. However, it's not clear whether that doesn't necessarily imply that it's the novelty of the target that is going to cause you problems in modeling. It may be the case that you have actually a really novel antigen target, that there's some exploitable motif in the epitope, which actually, although it's globally similar to anything you've seen previously, it's actually key for binding, and that one motif is all it takes for the antigens that would neutralize it to have a common structural feature with antibodies you've seen previously, and therefore modeling the important part is easy to do. And so in that respect, you might actually generate a really good model, even example hasn't seen antibodies for anything like it before.
You know, like I've seen in the SARS Plus example that you brought up, where we didn't have much data on SARS-CoV-2, but we had some data on SARS-CoV-1. And there, the homology of the, even though it was a novel target, the homology of the antigen kind of saved us.
Exactly.
Okay, we're going to take one more question because I think it's going to be really quick, and then I'm afraid we'll have to wrap up. No worries, though, for people whose questions did not get answered, the speakers will contact you with the answers. And that quick question is the structural annotation method species agnostic?
Yes, that's, you have a sequence. You can number the sequence, pick out this is the framework region. These are the sequences for all the sequences. So and then being able to do for example, a database search for sequences or parts of sequences that are homologous to what you have. That's all completely species agnostic. So you can apply it to sort of sequence from any species. Again, coming back to this idea that all of these methods are data-driven structural data we have. Obviously, some species are incredibly overrepresented in the PDB, human and mouse being the obvious ones. So if sequence on mouse sequence, your structural annotation method is on average going to yield better potential than if you have a sequence from a very understudied species. But this is nothing to do with the annotation method itself. It's purely a consequence of being data-driven and the data sort of very, very imbalanced.
And we've benchmarked this at least on humans and mice who've tried holding all the mouse data and seeing if we get a better prediction just for human data. And what we found is feeding in all the data helps. It doesn't matter if we actually truncate it by the data that we end up in isn't worth it. So it's better to include both of them. So great question.
All right. Well, thanks very much. And again, I'm sorry we can't get to the last couple of questions, but no worries. The speakers will contact you with the answers. So in concluding, I'd like to thank our speakers for relating their insights into how structure prediction can enhance antibody repertoire sequence analysis. I thank you for joining the webinar today. An on-demand version will be available in a few days. I'll send a link to it via email to everyone who registered. Please do feel free to watch this or any of our on-demand webinars when it's convenient. Thanks again, and have a great rest of your day.
That's all.
Thank you.